My name is Dieter Wolf, and I'm the head of the RNA and Disease Laboratory at Westlake University. At the most fundamental level, our research is interested in how the genomic information that is present in our DNA is translated into the proteins that carry out the functions in the cell.
To a layperson, why is this important?
So basically, all of our cells, in order to divide, they have to replicate and duplicate their cellular constituents. And a lot of what is in the cells is protein. So in order for a cell to divide, it needs to produce the biomass that will increase its size to that level where the signal can be triggered that the cell can be dividing into two cells. And obviously where this is really important is, for example, in cancer, where you have uncontrolled cell division. So you could imagine this as like a push, that there is protein synthesis, or the making of protein is pushing the cells to grow to the level that they can divide.
Practically speaking, ultimately, what are you trying to achieve with your research?
With our research, we are trying to get the fundamental molecular knowledge that will allow us to understand cellular processes at a level of detail, so that we can understand that disease processes that subvert these fundamental cellular processes. And often the disease takes its origin in a mutation or a variation in the genome, which then disrupts a process somewhere downstream. And we are trying to understand that the chain of events that leads from the genomic change to the cellular change that is causing the disease. Ever since I have been a medical student, I was intrigued by cancer. Cancer is a menace to humanity. It affects everyone from small children to people in their prime time of life, and of course also the elderly. And we have a lot of elderly people in all over the world. So cancer is a very complex problem, and it's hard to study it comprehensively. But we thought one possible approach would be to settle on one of its fundamental properties, and that is uncontrolled cell division. So there would not be a tumor mass if there was not excessive cell division. So in order to divide, a cell needs to reach a minimum size, and that there is genetic circuitry that controls, or that creates a dependency of cell division on cell size. So in other words, there needs to be a minimum cell size for a cell to be able to divide into two. And in order to grow to that minimum size, cells need to replicate their cellular contents. And a lot of that content is protein. So we thought that one way of looking at that problem is to try to understand how the cell creates biomass and protein. And now protein synthesis has been studied for many decades. And it's in some ways a simple process, because what you need to do is to put the individual building blocks of proteins into a linear chain. The building blocks are the so-called amino acids, and there are simple chemical peptide bonds that need to be formed from those building blocks in order to make the linear protein chain. So all of this is chemistry, and that chemistry is carried out by enzymes like most things in the cells. But what we have been learning is that this is not the end game. Once you have the linear chain of amino acids coming out of the machine that puts this together, which is called the ribosome, then there is a whole other series of events that need to happen in order to make the protein function and carry out its cellular function. For example, the protein has to fold in its three-dimensional structure, and also it has to be sent into the right places in the cell. The cell is not a sort of a bag full of mixed contents. It's a highly structured environment, and diffusion usually does not get proteins to their destination. So there's targeting mechanisms. That's another thing we study, and that is tightly integrated with the process of making the protein. Making it alone is not sufficient. It needs to fold correctly. If it folds, it does not fold correctly, it can actually cause a disease. So it needs to fold, and then it needs to be sent to the right place. And even then, the story is not over yet, because it also has to be removed from the cells eventually. So proteins have a life, basically, and we are trying to understand it from birth to the end, mostly focusing on the birth and early life at the moment. But in some of our other work, we have also focused on the death and also on the life-death decision. And what I mean by that is that a protein, once it's made, there is a quality check. If it's functioning properly, it can live. And if it is misfolded or does not function properly, it has to die. So that decision is not a random decision. That is made by molecular mechanisms in our cells.
Why did you decide to come to China of all places?
You know, I've always, actually I'm from Germany, but I have worked in the United States for 25 years. After my medical studies, I went to Boston to do a postdoc, and then never left the United States until 2019. But so I started my lab in 1998, and always, that was in Boston, always had Chinese researchers in my lab. And often they turned out to be among the most productive, and so they have always greatly impressed me. They are still my friends. Many of them are here, Harbin, Shanghai, Guangzhou. And so for me to consider taking a position in China was not such a big leap. I had been interested in Asian culture for some time, mostly through visits to Japan, just a place that's easier to visit. But then in 2009, I think I was taken on a trip to China by a colleague from Harvard, and gave some talks, and so yeah, I became fascinated with the country and the beauty of it, and also enamored by the people. So in 2015, when I took a part-time position at Xiamen University for me it was not such a big leap. Of course, I had to adapt to many things. Unfortunately, I don't speak Chinese. And I'm not sure that I will be able to learn it. I'm looking back at 8 years in China now, part-time, but still, it's been a really successful time. And I didn't see a way to replicate that success in any other setting. During my further research about the university, of course, I realized that it had, despite its young age, already attracted a tremendous number of really very high-level, successful people. So that I knew would make for a very interesting and stimulating academic environment, and also create new opportunities for collaboration. And then, of course, in the further discussions, it became clear that the resources this university can provide are second to none. Both in terms of what is available as a shared resource to all faculty members, as well as what they can provide for building my own research group and doing the research I love to do without constantly being on the lookout for additional funding, or constantly worrying how I can do the next experiments. And I've been in that situation. So I really look forward to the next 10, 15 years of really unencumbered scientific creativity, and that's why I came here.
You've been in Hamburg for 3.5 weeks. What are your first impressions of the city?
The city is big compared to what I'm used to. I came in the middle of February, so just in terms of the vegetation and the landscaping, it was a bit dull, I want to say. But during the past couple of weeks, when it warmed up, of course, all the trees started flowering, and the magnolia trees, and so really the city has become a lot nicer. I'm lucky that I found a really nice neighborhood to live in. So that I'm settled. Everything is provided for, and I can now go out and explore the city more. I have been to the Westlake, of course. That is a stunning scenery with the mountains in the background and the tea fields. So yeah, I understand why people come here just for that, and I'm fortunate to live here, so I'm gonna make a lot of use of that environment. It's not been a long time yet, but I have no doubt that I will be comfortable here all around.
What are your goals here at Westlake University?
Well, I have sort of goals at different levels. I have personal goals. I want to enjoy my professional life, which I think I can do here. And so the joy in my profession comes from different sources. There is the people that I work with. So I'm looking forward to building a strong team of people who like to be together and do this work together and have a sense of mission. And another component is the answers that we get, or you could say the success the questions we answer, the work we present to the outside world, publications, yeah, that is the way we present things, presentations and publications. So that is what makes a scientist feel good, a sense of community, a sense of impact, a sense of recognition, for sure. And I have had a good amount of that. I'm lucky, thankful in that respect, but I don't feel saturated yet, let's put it that way. So I have some ways to go, and there is still some things I want to understand and some questions I want to answer. And so for the future, I think I want to expand more into the direction of human disease modeling. Our research so far has been mostly on relatively artificial system cancer cells that have been in a test tube for many decades, and they change in ways that we cannot predict. But I'm now becoming really interested in modeling disease from pluripotent stem cells, which is an area that is new for me, but luckily I have colleagues here at the university who have a lot of expertise in that. So that is going to be one direction. And then I'd like to understand more about how RNA, so that's the molecule that from which the proteins are made, how RNA contributes to disease, and also whether we can use RNA as therapeutics or as therapeutic targets. So those are my sort of initial ideas of the direction in which I want to take it. Right now we'll start out with things that we know and that we have important work to finish, but in the long run I want to expand into that direction. That's another reason why I came here, because no other place will allow me the freedom and the resources to, at my age, to venture out and do something completely new.